This is the second in our ACES Technology Showcase webinars. And for those of you who don't know, ACES is an ARC Centre of Excellence in Electromaterial Science. And we're charged with the discovery, design, development of new electromaterials, but the integration of those into structures using advanced fabrication in order to tackle challenges in energy and also in health. Now, today's technology showcase is very much focused on biofabrication and what we're pushing towards in the health area. And many of you will know that there's been tremendous advances in 3D bioprinting in recent years. The ability to distribute biomaterials in different types of cells in 3 dimensions by design, from what we can put on the screen into a real structure. One of our real challenges has been in recent years is how do we characterize that? How do we see what we've created? How do we know that we've got that elegant distribution of the cells and distribution of the materials? And then what happens afterwards? And of course, we need non-destructive techniques to enable us to do that. And so it's great that Andreas is going to showcase this new technology from ACES based on ultrasound, which tackles that very relevant challenge that's in front of us today, using a really exciting new approach. So I'm going to hand over to Andreas.
Okay, thank you, Gordon, for the introduction. Okay, so welcome everybody. It's a pleasure for me to introduce our Ultralmage device, which is a benchtop quantitative ultrasound image device for the quantitative ultrasound evaluation of bioscaffolds and tissue engineered constructs. The motivation of this work brings a solution of the Ultralmage to the need of non-invasive characterization techniques to follow the biofabrication process. As you may know, the biofabrication process uses stem cells, which are very precious materials that are combined with biomaterials and complex fabrication processes, and then they are maturated over time with the purpose of their mimicking tissues of natural origin, their eventual implementation into humans, and also they are used for the in vitro disease modeling or pharmacology discovery. And the need of Ultralmage arises from the need of imaging techniques in tissue engineering, which are mostly focused nowadays on microscopy, which is always preferred due to the high resolution that provides, but often requires destructive methods such as staining, histology, or optical cleaning protocols. And moreover, this technique is difficult to apply in highly scattering materials and also has limited penetration depth. An alternative to this is optical coherence tomography, which is well known for ophthalmology and is optimum for light scattering materials. Has greater penetration depth than microscopy, so this depends highly on the material properties. It has high resolution, however, it needs contrast agents in some applications since it's based on the refractive index. And usually is used for microstructure visualization rather than for quantitative purposes. An alternative to this is photoacoustic imaging, which allows greater penetration depth than microscopy as it uses infrared light. And resolution depends on the transducer properties, typically about 30 micrometer axially, and is characterized by high specificity, which based on the laser and the ultrasound interaction, but requires endogenous absorbers such as hemoglobin. That's the main use of photoacoustic imaging for probing vascularization and flow of blood. And when used in tissue engineering, will require the use of exogenous additives. And another popular technique used in tissue engineering is Raman, which is mostly used in 2D and histology sections. It is high resolution and is used for chemical labeling. However, again, as with optics, it has limited penetration depth. And an alternative to this is diffuse Raman scattering, for example, for the imaging of cartilage, but it doesn't have imaging capabilities. So back a few years ago, when we started planning the Ultralmage project, we identified ultrasound as a suitable technique for tissue engineering, as it has greater penetration depth than optic-based methods, is ideal for soft tissues with light scattering properties, It has a micrometer scale resolution and is non-invasive and label-free, and overmost is a versatile characterization technique that allows through transmission and backscatter measurements. So in both measurements, the sound propagation and backscatter are dependent on the tissue composition and microstructure. To give you a bit more background on backscatter, this allows the grayscale imaging, which is typically used for construct morphology, and more specific, another alternative, quantitative ultrasound imaging, which specializes our Ultraimage device, is basically the analysis beyond grayscale imaging, which is the frequency component analysis of the radio frequency signals, and allows power compensated and user independent images. And about through transmission, briefly, is you can measure the sound speed, which is related to the bulk mechanical properties of the constructs. And you can also measure attenuation, which is the sound dissipation by scattering and absorption processes, which in relation to tissue engineering, it can be related to the formation of extracellular matrix. And here you can see a few reviews of imaging technologies in tissue engineering and biomedical engineering. And if you look through, you will find very limited number of reports using ultrasound for tissue engineering. And we believe that this lack of reports are due to the absence of commercial products for carrying out this type of investigations. So researchers are limited to use modular systems, as in our case, or use clinical and preclinical systems that are difficult implementation, are not designed for this purpose. They are fantastic for many other purposes. They have been improved over decades, and they are really expensive. In our case, we have developed Ultraimage that is dedicated design for tissue engineering and dedicated software for tailored signal processing, and is affordable hardware. With this Ultraimage device, we can do several measurements that are useful for the non-invasive evaluation of biological constructs and biomaterials. Among those, we have used it to use the characterization of biomaterials in terms of thickness, doing morphological changes such as maturation or hydrogel degradation. We have used it to investigate surface roughness of biomaterials, back modulus, and hydrogel degradation. Another application, I am going to show you an example later, is the cell distribution and proliferation of cells, which can be used to study cell growth kinetics or investigate the effect of cell growth factors. Another important area of application is cell cluster size determination. This can be relevant for tumor cells development and can be used for screening of drugs against tumor cells, such as the efficacy of the drug and the shrinkage of those tumors, and can also be used for monitoring drug release when they are, for example, they are typically encapsulated into spheres, and they will shrink over time, and this can be detected by ultrasound as well. Another important application, and we are focused lately on this, is the extracellular matrix formation that can be used to monitor cartilage formation, bone, or dermal layer in vitro constructs. And last, we can use this technique for the structural evaluation of bioprinted constructs. So starting with examples, here's our first publication we did on the area of ultrasound, which we were correlating the hydrogel's sound speed and swelling thickness with the Young's modulus. Here we prepared a series of hydrogels of different swelling properties, and we were using time of flight measurements to determine the thickness and sound speed. And we published a modified equation for the back modulus to account for the hydrogel swelling. And we noted that this publication has been overlooked in literature, and we understood that not everybody has an ultrasound system, and therefore we also developed a method that you can follow the thickness increase after swelling and can be calibrated to the hydrogel Young's modulus. Another application is a big development of the Ultraimage was the use of Ultraimage for the quantitative ultrasound imaging of hydrogel bioscaffolds laden with cells. In this example, we use PC12 cells laden in collagen hydrogel. And here you see an example of the grayscale image, and on the right, the quantitative intensity image of such grayscale image. And this is power compensated and user independent. Furthermore, we can calibrate this number of cell density to the intensity image, and we can determine the cell density per pixel. This property was used to develop, to follow the development of PC12 cells over time. On the left, you see the cell density of cross sections such as this inside the hydrogel, and we use it to follow the cell growth over a 72-hour period of interval. If you are familiar with PC12 cells, you will know that these kind of cells are kind of tumor cells, and they grow in clusters. And this is exactly what was captured by ultrasound, where with the progression of time, you can see a shift to red pixel index in clusters as represented in the optical image. With the use of quantitative ultrasound imaging procedures, we were also able to model the cell cluster size out of the ultrasound backscatter spectra. And again, we were able to determine the cell cluster size over time. Another feature to point out is that with the use of precision stage controllers, we were able to record the progression of cell growth over the same spatial point over time. And this could be used to study the kinetics of tumor development. And here to give an example how we model the cell cluster size is by the analysis of the spectrum that we collected, and we modeled it with a theoretical model. And here we found the best agreement, which corresponds to a 98 micrometer diameter, which is also corresponds with the optical images. The next big achievement in Ultra image development was the integration of 3D image capabilities, and in this case showing an example for interrogating the internal structure of bioprinted constructs. You can see on the left the bioprinted construct of 10 by 10 millimeters and 2 millimeters in thickness. And with the use of ultrasound, we are able to go through the layers and determine and verify that the number of layers of the construct correspond to the initial CAD model. This can be used by researchers to optimize their printing parameters, and when integrating cells to follow the development of cells into this 3D construct. The next big achievement was the integration of 3D approaches. 3D approaches for the evaluation of bioscaffolds. In this case, we are focusing on measuring and quantification and the distribution and quantification of extracellular matrix in these bioscaffolds. And we do this by generating quantitative ultrasound images over the construct. We patch them together into a 3D image. And here we can generate the 3D image. We can look at the morphology of the construct in different orientations, and we can also represent at different intensities to visualize the inner microstructure as well as the density of this extracellular matrix. Furthermore, we can also generate a virtual slice of the construct, which is different to the orientation that it was measured. And from here, we can do a color analysis on these 2D slices or volumetric analysis to determine volumetric properties of the construct. And lastly, we can also look through the construct and generate these kind of scans that are called contour scans, that we can read out the image of the construct as well as the sound speed for example, among other parameters. This project was used in this example together with the collaborators from Melbourne to analyze the distribution and quantification of extracellular matrix in this kind of bioscaffolds with differentiated stem cells to produce neocartilage. And as I mentioned, we have used this method to look into the gel and understand the extracellular matrix density. Together with the collaborators, we have prepared a range of constructs where we have modulated the chondrogenesis by changing the hydrogel composition, namely from soft to hard. And we have performed an extensive analysis of the images, and we have determined that this yellow index intensity is the most relevant for the following chondrogenesis, and this has been correlated with biomarkers. Another thing to highlight is that these samples have been prepared in Melbourne and have been sent by mail to Wollongong as a fixed examples, and this validates and this demonstrates the possibility to collaborate with groups that are far away. Another example of using ultrasound to follow extracellular matrix formation has been applied in cell spheroids. In this example, we have used the technique to measure the same cell spheroid over time over a period of 28 days. And you can see the first column, we show the sound speed development into the construct which we attribute to the extracellular matrix formation. And you can see from pale blue into brighter blue, indicating this densification of the extracellular matrix into the cell spheroid. We can also look at the development of the morphology, which after 21 days remains fairly constant, and then at day 28 makes a boost into the diameter due to the maturation of the differentiated cells. And lastly, in this column, this is very unique of ultrasound that allows you to view the inner structure of a really opaque material, which would not be possible to determine by other methods. And here you have an image of the cell spheroid that was measured of about 1.38 millimeters in diameter and was measured in this configuration. And just to highlight, the quantitative ultrasound imaging does not interfere with cell activity. It can be applied to opaque constructs. It can reveal the inner microstructure, which otherwise is inaccessible by other methods. And we correlated with bioassays. So what's next? We would like to continue with the national and international collaboration. We would like to explore more ultra-image applications and understand researchers' needs for further development. We would like to develop a professional software for distribution and commercialization for those who need it. And finally, we would like to design and build a standalone system. Finally, I would like to acknowledge the ARC Center of Excellence for Electron Material Science for the funding and support, as well as the ANF Materials Node for the equipment use, and special thanks to the collaborators from Melbourne University, Carmine, Serena, and Claudia, and here in Wollongong, all the people that have helped me in the publications or in any other means during the development of UltraImage. And thank you for all. If you have interest in collaborating for us, please send us an email to either me or Gordon. Thank you.
Okay, we can stop sharing and see if there's any questions. Okay, great. So if you have any questions, you can just put them in the chat box, or we don't have an unruly number to deal with here today. We've kept the numbers on these low. So you can also just turn off your microphone and ask us a question if you like. So if there's any questions, put them in the chat box or just turn off your microphone and ask a question. So while you're thinking of that, I'll just ask Andres a question around, so what's the typical length of time required to get those three-dimensional scans of those gels or the 3D printed structures?
Yeah, that's a good question. Well, it can take several minutes. Depends also on the resolution. So the transducer has a lateral resolution of 120 micrometers. So you can decide to do side-by-side scanning or overlap, and each scan takes about 1.5 seconds. So if you want to do a 4x4 millimeters with 80 micron resolution, this will be about 3,000 scans, and this will take you about 90 minutes.
90 minutes, okay. Is there any other questions? As Andreas mentioned, we have collaborated closely with our colleagues in Melbourne on some of those later aspects of the work, and I see that Serena and Carmen are on screen with us today. So I might just invite them if they have any comments on the system as to how useful it's been in terms of getting information in this non-destructive way. So I'll flick over to Serena and Carmen if they have anything to add.
Yeah, so thank you, Gordon and Andres, for the presentation, and thank you for all the work done.
Okay.
Because, yeah, we really think that this technique is revolutionizing our way to analyze the 3D bioprinted construct. For us, is really relevant for the cartilage regeneration. So we do heavily use gelatin-based hydrogel with stem cells. And during those years, we have developed techniques to actually analyze what is happening inside the samples, but all the techniques requires the destruction of the samples. So we can't really follow the same samples over time. And for us, it's really crucial to understand what is happening during time, because we are dealing with differentiation of stem cells. So we really need to understand if the cells are really doing whatever they are required to do. So the process of neocartilage generation is a long process. So we are talking about weeks of, at least in vitro, and it's even longer in vivo, of course. So at some point we needed to speed up the analysis, because even the analysis, they take quite a lot of time. So the classical histology, it takes even a full week, okay, of processing. So, and again, we are talking about destroying the sample. So, and you can imagine all the artifacts that you can that you can generate, and so all the results that can be false results, or so you need always the proper controls, and you need also in terms of number of samples, you need a lot of replicates, because, yeah, as I said, you can't really follow the same sample. So we also tried other alternative techniques, such as light sheet imaging. But unfortunately, so the degree of penetration in those samples is not great, and especially when the cells are starting to produce the neocartilage, so the samples become really opaque. So the limitation on the limits of the optics, so you can't really penetrate farther than microns. And instead, we are dealing with samples, as Andres explained, of millimeters of thickness. So yeah, to wrap up, I think that this is a really great technique. And also, if I can add something for the future development, I think especially for the in vivo studies, because of course we are doing also translation in preclinical animal models, and hopefully real soon in humans, or in clinical trials. I think that the ultrasound being established also as a technique for other applications in humans, it can also be applied now for following the regeneration in this construct inside a human being or even an animal. So I think that I'm really looking forward for the future development and for the future application for the translation. So we'll see.
Great.
Thank you.
Okay, great. I've got a couple of, thank you for that, Serena and Carmen. And if you have any questions around the work specifically on the cartilage, please feel free to also just email Serena or Carmen directly down at St. Vincent's Hospital, University of Melbourne there. And we'll just move on to a couple of other questions that we've got here on the chat box around. The first one, Andreas, is around the software you use to build the 3D images.
It's all done with MATLAB. Yep.
Okay.
Yep.
That's an easy one then.
Yep.
And this is an interesting question. Could this ultrasound be used to distinguish between different types of cells?
Well, no, not really. This is one of the lacks of ultrasound. It only measures the amount of scattering inside the sample. So unless the two cells are behaving differently, you will not be able to detect, to distinguish.
So it doesn't differentiate between the cells. What about the artifacts from boundary conditions such as reverberation? Do you encounter that, or do you prevent it or control it?
Well, there is a artifact when you measure close to a surface, like a hard surface, like glass, you will see interference echoes. That's why we are using a cling film and at the bottom a polystyrene layer. In terms of artifacts around the surface of the hydrogels, there is some, but to a minor extent. And with the analysis we perform, with the volumetric analysis, all this is factored in, and it is still useful to measure biological processes.
Great. And what about, you did mention about measuring the degradation of a hydrogel, I think, using the technique. How does that work?
Well, basically, sound speed depends on the density of a material and the mechanical properties. So if you have a hydrogel that is degrading, its density and structure will be altered, and you can register this with sound speed. You will see a decrease in the sound speed.
And this is sort of related to your resolution and scanning time. I think you spoke about before, monitoring things as a function of time, such as cell migration through a matrix, given you have a non-destructive technique.
Yeah, it can be tricky if it's a well-saturated sample, but if you have, let's say, one half of the hydrogel is empty and the other is with cells, you will see definitely a change. You will see the migration of cells into the other side. Yes.
And I guess the resolution on the time axis there then is the time taken to completely scan the image, right? So
exactly.
it's minutes.
Well, if it's one plane, you can do a fairly quick image, just a few seconds. It takes time when you want to do a full 3D image that can contain thousands of scans, like 3,000 or 4,000 scans.
Right. Okay. Okay, great. Are there any other questions, either directly, if you'd like to just turn your microphone off and ask, or thank you for all of those questions in the chat box. That's great. It's your last chance to grill Andreas. He's in the hot seat.
I'm just happy to present the device. Thank you. Thank you for listening.
Yeah, so look, from me, thanks everyone for dialing in and having a listen about this exciting new technology that Andreas has pioneered. We hope we get more questions from you in coming days. This is just the start of the conversation. And look, the whole purpose of these technology showcase webinars is to identify and establish new partnerships that can help us accelerate the translation of the technology. So if you have any interest in doing that, whether that's as a user of the technology or some ideas around how it could be improved, around the software development, the instrumentation development, or any commercial expertise, I know that Andreas and I would be very happy to hear from you, and I hope that we do over the coming days and weeks. So thank you all for dialing in. A huge thanks to Andreas for putting together the presentation. It was excellent.
Thank you.
And also to our colleagues from Melbourne for helping us at the last moment just to get a user's perspective on the Ultra image. And we'll see you all again sometime soon. So thank you and bye-bye.